These couple slides will explain the lactate shuttle and lactate threshold, and also some updates to the lactate shuttle hypothesis. Some of these notes and slides I've taken from the ACSM Advanced Exercise Physiology book. So the goal of this video is to look at glycolysis and look at the lactate threshold. And we've seen this picture before, a classic picture of the lactate threshold, whereas in this case treadmill speed increases, there's an increase in blood lactate concentration. And where there's an exponential increase, where the curve changes shape, this is classically defined as the lactate threshold. And this is a typical view. So what about energy systems and energy systems that would be producing lactate? So the three energy systems, the direct phosphorylation of ADP to ATP, that immediate energy system, glycolysis, breakdown of glycolysis and ATP production, breakdown of glucose, and the aerobic or long-term energy system happening inside the mitochondria. And an important note is that these three energy systems are happening simultaneously, not sequentially. So even though the ATP phosphocreatine energy system is classically defined as immediate, one is still using some of that energy system, along with an aerobic energy system. So it's based on the ATP ACE demand. So the control of each of these energy systems depends on the regulated response of the whole system to the change in the rate of ATP ACE activity. So here's an overview of glycolysis from a picture I've copied, and of course it's the breakdown of glucose and the formation of 2,3-carbon pyruvates, or if we're looking at lactate production, those pyruvates would then form lactates. And the classic view of this, if we break down the top part of this picture, it's the energy investment phase. And the only things I would like you to be aware of are these enzymes over on the right-hand side. So be aware of the first enzyme, which is hexokinase, and that is the enzyme that will phosphorylate glucose to form glucose 6-phosphate, and it's energy dependent, so it requires an ATP. And then step number 3 is the enzyme phosphofructokinase, or officially phosphofructokinase 1, and that also is energy dependent, and it's the formation of fructose 6-phosphate to fructose 1,6-bisphosphate, so adding another phosphate group onto this. And this is the preparatory phase of the energy investment phase, and this is the payoff phase or the generation phase. And here the enzyme to be aware of is this last enzyme, which is called pyruvate kinase. So it's taking this phospholinopyruvate and making pyruvate. And later on in these slides, I'll talk about one additional enzyme. And obviously ATPs are formed, and NADH, electron carrier molecules, are formed. And so this glycolysis pathway, it does not require oxygen, so it's anaerobic, and I like to call, and others like to call, this fast glycolysis. It's happening relatively quickly. In terms of ATP production, it's a net of 2 ATPs if we started from a single glucose. And the enzymes to be aware of are hexokinase, phosphofructokinase, and pyruvate kinase. And be aware that the end products are NADH, and pyruvates are lactates that could get transported into the mitochondria. And just so you're aware, a kinase enzyme, so hexokinase, phosphofructokinase, and pyruvate kinases. Kinases are enzymes that actually move they move or transfer a phosphate group from a high energy phosphate like ATP, and they will donate those to the substrate like glucose to make glucose 6-phosphate. So this process is phosphorylation, adding a phosphate group to something, and the substrate gains a phosphate group, and the high energy ATP will donate that. So kinases are transferring or moving this phosphate group. And the end products of this fast or anaerobic glycolysis are lactic acid or lactate. And once lactic acid is made, it's almost immediately the hydrogen ion is removed, and the anion lactate is what is left over. So this lactate threshold. So what are the causes or mechanisms of this lactate threshold? And typically there are thought of 4 mechanisms that can cause this lactate threshold. It could be low muscle oxygen. It is an accelerated rate of glycolysis. It is a recruitment of fast twitch fibers, and it's also a reduced rate of removal of lactate from the blood. So this lactate threshold, again, the graph is an exponential increase in blood lactate concentration, like millimoles per liter, as exercise intensity increases. And I like to think of this blood lactate with this simple diagram of a rectangle. And the middle of this rectangle represents lactate in the blood. So that's the concentration of lactate in a given volume of blood, like millimoles. And the 2 arrows actually represent the rate of lactate appearance in the top left, and then the rate of lactate removal down in the bottom right. So what we are measuring and what we can measure in our laboratory is the concentration and the lactate threshold that we saw previously. It's simply the concentration of lactate in the blood. And that's all we can do. Yet that is limiting. That measurement is limiting because we don't know the rate of appearance and we don't know the rate of removal. And actually at steady state exercise or rest, these rates are equal. And it does not mean that we're not making any lactate. It just means that the rate of appearance of lactate is equal to the rate of removal. So this is a very simple but a good diagram to sort of think about this lactate threshold and what could be causing this lactate threshold. So this first video was talking about glycolysis, and then a little bit about the lactate threshold.